Good time of day, folks. This is Sharky Prodiver, and welcome to the IB Survivor Diary. As you noticed, I don't wear my hat today. It's because I wanted to show you the mess I've created after washing my hair and applying the, how do you call it correctly, the hair dryer? Yeah, the hair dryer to it. It kind of looks like Wolverine stylish, except Wolverine has got more awesome hairstyle. So I think much better. Alright, the topic of today's video is not about preparing for an exam or talking about sorry for that, or talking about my social life. The topic of today's video is discussing a test. I got my first biology test back and I'm at the same time happy and frustrated. I'm happy because I got a fairly high grade. I'm frustrated because I was 1 point off a grade boundary. Now, I was expecting to lose at least 2 points in my test. I ended up losing 16. And about the grade boundary, I got 34 points, which is 68%. Had I got 35, which is 70%, I would have gotten a 6 out of 7. But now I stuck up with a 5, which kinda added an additional kick down to my grade experience. So I decided to look at my test, tell about the questions, tell what I answered, what I should have answered, why did I do it wrong, and what to do better for the next time. Let's get started right in. To give you an introduction, the test was given on 4 topics. The first 2 topics were called Introduction to Cells and Origin of Cells. The second topic was called Ultrastructure of Cells, and the third and final topic was called the membrane structure. Now that you know what topics are in the test, we can at least have an idea of what I'm about to discuss. There were, let me check correctly, there were 7 parts to this test. The first part consisted of multiple choice questions. The second part was talking about the cell structure and membrane. The question 3 was about emergent property. This question 3 was about emergent properties of the cell. Question 4 was about surface area to volume ratio. Question 5 was about the function of membranes. Question 6 was identification of cells. And question 7 was about the stem cell therapy. Let's start. Well, I'm not gonna cover too much time on my multiple choice question. I will only say that I got several things wrong, such as I wasn't able to correctly identify the functions of the claims that support give me a moment. Control Z F5. I wasn't able to correctly identify the claims that are supporting the cell theory. I did mess up the magnification, and I kinda messed up the membrane proteins, just to be generic, and also the question about the origin of cells. Now then there was the question 2, and I was given that micrograph, and I was supposed to study it, and I then say what cell is presented and why. And as you see, the present cell is a eukaryotic cell. Why? Because we can see that there is a nucleus in the cell, and we also see the presence of many organelles in this cell, such as the endoplasmic reticulum and a few mitochondria, and so on and so on. Then I got a question that was basically the very same picture again. Can you see it? Yeah, like that. The very same picture again, and I was to identify at least 4 organelles there. I basically said nucleus, nucleolus, lysosome, mitochondria, and then I said rough endoplasmic reticulum. So I said 5 just in case, because if I get something wrong, which I actually did not, which was good to hear. And then I had to draw a diagram of the membrane system labeled A on this photo. As you can see, this is the A label, and it shows to the nucleus. Now, the trickiest part about the membrane is that what you need to do in every membrane is to show the phospholipid layer, so with the hydrophilic side of the phospholipid facing outwards the membrane, and hydrophobic facing inwards the membrane. Then you need to show the protein types, the transmembrane protein going out through all the way through the cell, peripheral, and what was the other one? Jesus Christ, I forgot it. Shame on me. So peripheral, transmembrane, and integral protein. I forgot that. Idiot. I also had to show, because it was the nucleus, and nucleus is one of the three structures, chloroplast and mitochondria being the other two, that has a double membrane. So I just don't show only this part, I also draw the second layer of the membrane to show that that's a nucleus. And that's the tricky part in the question, so that was actually quite a surprise. Then I basically had to state two functions of the endoplasmic reticulum. Those two functions were quick and effective transportation of chemicals for use in and outside of the cell, and the second function is attachment point for ribosomes in order to generate proteins for use in the cell. Well, then there was question 3, and question 3 is something I completely and utterly messed up, because it asked me about the emerging properties in the cell with an 8 example. So what I had to talk about was to say how over time certain functions in the cell stayed and how they disappeared, and also because the question asked me about multicellular versus unicellular organisms, I was to say how come multicellular organisms are more efficient than unicellular, and I was, for example, to name the respiration, a process that requires a significant amount of organelles, however that also provides a significant amount of energy. Guess what I did? I talked about the endosymbiotic theory. And endosymbiotic theory is completely and utterly different from the emerging properties in the cells. Why? Because endosymbiotic theory talks about how back in the time there was one cell that got two other cells bumped into it and they formed together and started functioning as a single unit. That's kind of related, but that's not what was asked to me. And then to top all that off, I started talking about how different organelles do different functions, which, as you see again, is off topic. So I got 0 points for that, and when I got my test back, that was basically my reaction. But well, we will learn on mistakes. The question 4 was about surface area to volume ratio, and that I did good. I got 6 out of 6 points. Basically, you were given two cells, one big cell, one small cell, one big cell, one small cell, and you were given also their surface area and volume, and you were to determine the surface area to volume ratio for both cells and deduce on the results you received why cells stayed small during the process of evolution. What you get in the end is that the smaller cell has larger surface area to volume ratio. That means it can allow more reactions to happen on the membrane. It means more stuff can go in, more stuff can go out faster. That's it. The question 5 was a tricky little sneaky butt rape question. Why? All it asks you is tell 4 consequences of a membrane dysfunction. I went all the way everywhere. I talked about how good cholesterol goes away from the membrane, becomes bad cholesterol, which is chemically false because good cholesterol and bad cholesterol have 2 different formulas. I talked about how membrane breaks down or doesn't break at all, therefore cell cannot be divided, etc., which is not really what they were asking me. What they were asking me is quite simple. There are proteins on the membrane that do different functions. And in the syllabus, we were given 4 specific functions of the proteins on the membrane of the cell. Basically, transport, communication, activation, and binding. And that was basically to state how those 4 functions were to be affected. If, for example, the proteins on the membrane were to go down. That's it, but I was smart. I decided to go somewhere else to do some rocket science. Kids, never do rocket science when you don't need to. Then there was question 6, which was easy peasy. I was given that thing, and I was asked what that thing is and why do I think that that thing is plant or an animal cell. I don't even know what that is. I think that's an animal cell. I'm just joking. Of course it's a plant cell. Why? Thick wall, big vacuole, presence of chloroplasts. Easy. So that was a question I was able to answer with the more or less certainty, which kind of gave me feels, positive ones, to keep on going. And then there was the last question, question 7. Question 7 basically asked me to discuss the techniques used to generate therapeutic stem cells. Now, there is embryonic stem cells and therapeutic stem cells. Embryonic stem cells are stem cells taken from something that was never alive, so from an embryo. Therapeutic stem cells is cells taken from a living organism. So, like, if you take stem cells from me, that's therapeutic. And I was basically to outline it, that's to say that, for example, the DNA is taken from a person who needs the stem cells and then injected into the empty egg donated by a female donor. Egg is then developed into a blastocyst, and the stem cells are then extracted and put into a petri. Oh my god, sorry for this. And the stem cells are then extracted and put into a petri dish where they are then shocked to be created into a specific type of cell, such as nerve cell, eye cell, skin cell, etc. And after the sufficient amount of cells is grown, the piece is taken and put back to the patient. That's what I had to say. That's what I basically said. The only thing I did not kind of put down well is to say that the egg donated is empty of DNA. I didn't say it was empty of DNA, I just said it was an egg donated, and this actually makes a difference. The last question was to discuss one ethical issue for and one ethical issue against, and the therapeutic stem cell research. And basically the positive ethical issue is that if you take my DNA and you want to transplant those cells to me, there is a very small chance of rejection. So you can almost guarantee that if the operation is done correctly, that my body will not reject those cells. The negative issues, for example, cloning, because it's an ethical dilemma. Are we allowed to do clones? Are we not? Is it good? Is it bad? And basically I stated that. Now, on to the marks. That test was out of 50. And those are my marks. So, for the multiple choice, I got 5 out of 10. For the question 12, I got 12 out of 14. I got 0 out of 5 for question 3. Maximum for question 4. 2, I think it's 2 out of 4 for question 5, maximum for question 4, and 5 out of 7 for question 7. So now you know the results, now you know what the test was about. The question is, why did I fail certain things? Why was I good at other things? And I actually decided to analyze it, and the answer actually was pretty damn simple. The answer is that during my preparation for the test, I studied topic 1.3. which is the membrane structure. I studied the topic 1.2, ultrastructure of cells, which also entitles the surface to volume ratio. But I did not study those, because on the day when I was studying for it, I did not take it with me. I left it at school and then I took it later and just did a quick recap. So why was this a problem? Because this was the first topics that were discussed, so they were the longest time ago that I saw them. This means I had other things to remember and I forgotten a part of it. And I did not rehearse it correctly. If you actually look at my tests again, the multiple choice questions were primarily based on topic 1.1 and 1.5, as well as question 3. And you can see that in those areas I failed. 5 out of 10, 0 out of 5. That is the result of me not preparing enough for those topics. For example, what about the question 5, which is the membrane functioning, which I studied? In there I also neglected the proteins because I did not quite understand the topic and I was more focused on remembering the correct membrane drawings and the phospholipids and the topic 1.2, which is the structure of cells. So in conclusion, I prioritized and forgot some of the topics from my syllabus for the test, which in the end led to me losing those 16 points. quite precious 16 points, which caused me to not get a 7. So, what is the advice that I can give to other people so they can learn on my mistakes and do not make the same things? I advise to actually, when you rehearse for something, first of all, always take your notes with you, so you can rehearse. Second of all, when you rehearse, start with the earliest topic, rehearse it, and then go for the others. Third, don't prioritize. The notes are given to you by the teacher, that's the syllabus, so you should learn from all the notes. In fact, I actually quite liked the experience about the test, so despite the fact that I still, failed in all of those aspects, I at least now know why I failed it. And I think that I will be able in the future to make sure that I will not do the same mistake again. Well, that is all for my evening rant. That was Sharky Prodiver, and I hope you guys enjoyed this vlog. Have a nice evening and see you next time.